Hello. So my presentation is about computational simulation and biomedical application of two-dimensional photonic crystal hydrogel. So first, some background information. So what is a photonic crystal hydrogel? The photonic crystal hydrogel is composed of two materials. One is photonic crystal, another one is hydrogels. Photonic crystals are materials are similar to gratings. They have dielectric constants which changes periodically along the axis. Hydrogels are elastomeric in nature, so it changes when we apply pressure or stimuli or hydration. So transparency is a major feature of hydrogel. So if the material is transparent, we can use it for optical sensing. Here are some examples of naturally occurring photonic crystals and their SEM and TM images, and some representative images of the one-dimensional, two-dimensional, and three-dimensional photonic crystals. So the first question was, can we build a computational model based on the interference and mean scattering theory, which can be used to predict the diffraction pattern of the photonic crystals? So why do we want to build a simulation? If we have a simulation, then we can actually use it to relate a unique arrangement of particles and diffraction results and predict the response of a photonic crystal sensor and optimize the sensor for application. So here are we have the unit cells of different configurations and their latest geometry, which shows different nearest neighbor particle spacing for different configuration. I have three different configurations, for example, two-dimensional array, centered, square, and hexagonal. So how the simulation actually works? So our simulation has two planes. One is photonic crystal plane and observation plane. Photonic crystal plane has all the particle arrays, and observation plane has the observation point. So for each observation point on the observation plane, we do the following. So first, for each particle in the photonic crystal hydrogel, we calculate the path length distance and phase shift. From there, we calculate the electric field contribution, and we multiply that with the mean scattering factor. So each particle provides this factor. This ultimately was done for each of the particles present on the photonic crystal plane. And finally, we calculate the final intensity for each observation point, and which is repeated for all the particles, and finally to create a diffraction pattern on the observation plane. We have different configurations. So from different configurations, we simulate the array structure, and also we see the diffraction pattern generated from different structure. So we are applying conditions like perfect Sprague crystal, macroscopic error, and macroscopic error. You can see for the macroscopic error, it creates a dividing like structure. So to verify our simulation result, we compare it with the experimental literature provided diameter and also theoretical diameter. Our simulation provides very close diameter with the experimental literature and theoretical measurement. Here, because when we fabricate a hydrogel, we don't have a perfect perfect crystal. So it is always a combination of hexagonal or 2D structure. So our simulation we used to see what kind of combination provides what kind of dividing. So here we are showing different diffraction pattern for different mix of configuration. So in conclusion, our simulated result matched with the literature result and also the theoretical results. And so our simulation is not perfect. The geometry it provides, it's accurate, but the intensity, which we calculate because it's based on the single sphere mean scattering factor. So, and we have multiple spheres present in the photonic crystal. So it's actually cannot provide the intensity correctly. So it is uncertain, and currently we're working to make the model better. So next question was, can we fabricate a gel with fluorescent particles, and we can, so that we can use a conventional fluorescent microscope to analyze its structure. So why do you want to use a fluorescent-based microscope? Because when traditionally it's done with the ACM microscope, where the ACM image requires us to process the sample for vacuum chamber, requires dehydrated sample and speed-train technique. So after those processing, we cannot use the sample for further studies. So we want to use the fluorescent-based monitoring, so we can use the sample for further studies, for example, for dividing analysis. So here is a summarized version of my analysis. So we create the sample, fabricate the photonic crystal. We use it to measure, produce dividing. So we get an experimental dividing, and then we extract from the confocal microscope images. We extract their particle positions, and then use those. The gap of the particles is used to build simulated dividing and also the theoretical dividing. So here is a brief description of how the gel is fabricated. First, we use a polystyrene particles. We clean the particles, and then we use the ultrasonicator to prevent the aggregation of the particles, and use the particle with resin to increase the ion exchange. And then we inject this particle suspension inside a chamber where we remove all the above solvent. And once the photonic crystal structure is formed, we inject the pre-gel solution and cure it to collect the photonic crystal hydrogel. Simple schematic diagram for the experimental setup. We have the laser source, which we shine on the hydrogel, and dividing forms on the observation screen. We use a smartphone to capture the dividing. So we use confocal microscope to image hydrogel. The A picture shows the multi-panel mosaic image. B is the zoomed in picture, and E shows the centroid to identify each particle present inside the hydrogel. C and D shows the organization of the particle inside a gel. So B shows the experimentally generated dividing. A shows the simulated dividing, and here I show different. So it's pseudo color applied to each particles to show what kind of configuration they have. For example, the green one has hexagonal configuration, and red one has 2D array configuration, and blue shows random error that are present, which cannot be classified in either 2D or hexagonal. So we compare between the group of the dividing diameter, experimental, simulated, and enhanced theoretical. So 7 out of 9 samples shows there were not significant difference between the samples. So it shows similar dividing. So fabricated hydrogels were successfully analyzed using confocal microscope, and we show there are not significant difference in the dividing diameters. So the third question was, can we use this hydrogel to sense ultrasound pressure? Why do you think it can sense ultrasonic pressure? Because ultrasound has a longitudinal wave which is composed of compression and rarefaction. So when it comes across to the photonic crystal hydrogel, it creates a transverse waves which is perpendicular to the wave propagation. So that causes a displacement of the particle inside the hydrogel, and that results in the diffraction pattern change. So before we move to the testing the sample with the ultrasound pressure, we test the sample under physical pressure, to see if the dividing changes its size based on the applied pressure. So we find that applied pressure causes the dividing to shrink, and increasing pressure is the relationship is shown in the graph here. So this is the experimental setup where we use the ultrasound, and we have the photonic crystal chamber holder, photonic crystal, and then laser light and ultrasound is aligned in a way so it hits the same spot of the acoustic sound. So, and the light hits the same spot, and then the change of the diffraction pattern is collected by the photodetector. So the transducer receives a more output, and that is the transducer generated signal is verified and quantified by the hydrophone, and optical signal generated by the photonic crystal is collected with the photodetector. And here we have the same frequency, shows that the photonic crystal is actually capable of responses to the frequency generated by the transducer. So we tested 3 different transducers, 2.5, 5, and 10. So lower frequency shows there is a low pressure sensitive. It can sense lower pressure, and then higher pressure required for the high frequency transducer. So here we also show the limit of detection of the gel. So in conclusion, we demonstrated to be sensitive. Photonic crystal hydrogels were demonstrated to be sensitive to the both physical and ultrasonic pressure, and we have tested three transducers, and all were produced measurable response in the diffraction pattern generated by the photonic crystal. So that's the conclusion. Thank you.